So today I'm going to be presenting some work that I completed as part of my master's degree at Georgia Southern University, looking at just trends of seasonality in parasite community composition in shrews. So there's evidence that environmental drivers that vary seasonally contribute to the abundance and distribution of parasites and community composition in populations of hosts. And these things include precipitation, rainfall, temperature, things like host behavior, reproductive status, age, all of which are known to vary seasonally, right? But a lot of these historically predictable seasonal patterns are now experiencing deviations from these long-term norms, which make them increasingly difficult for us to both characterize and predict in the future for understanding like how we might expect communities of parasites and hosts to respond to a changing climate. And so some major goals that are associated with this problem are characterizing historical distributions of hosts and parasites as they relate to seasonality, understanding mechanisms that shape those distributions, and also being able to anticipate the ecoevolutionary consequences of those seasonal patterns. It's probably too late to do the first one, right? But we're gonna try anyway. And so an excellent host system for investigating this problem are shrews, because they're super small, they're abundant, they only live about a year, so if you're studying parasite dynamics in those host populations, you can have some more confidence that what you're seeing is a result of seasonal mechanisms as opposed to just infections that carried over from the previous year. They also have super high metabolisms and can starve to death in a matter of hours, which means they need to constantly be eating macroinvertebrate prey items, which results in constant exposure to parasites and some really diverse communities of helminths. So the two host species that I've investigated for the study are Sorix cinereus and Sorix monticula. They're closely related, they occupy similar niches, and in terms of the parasites that infect them, cestodes are the most common. And we've got 39 known species of cestodes that infect shrews, and 27 of them are documented as infecting shrews in the genus Sorix. So if you are a specialist parasitologist and you're not up to date on your tapeworm life cycles, an example host here would be a shrew, right? They shed tapeworm eggs into the environment. Those eggs are eaten by an intermediate host. Those eggs hatch and insist in the intermediate host, and they're then eaten by the definitive host, and this process starts all over again. And we can see that components of this life cycle are exposed to the environment. We can see that components of this life cycle take place in environmentally sensitive intermediate hosts. And so this sets us up to sort of ask questions of the system like, do tapeworm communities in these populations of shrews undergo seasonal change? And two, how do different traits that might be attributable to a host or the environment, how do they influence changes in parasite community composition over time? And so to sort of get at this problem, a team of researchers from University of New Mexico collected a crap ton of shrews, 182 of them, over several months spanning from May-October in 2016 and 2017, April and May in 2018, and September in 2019 across two creeks that are pretty comparable to each other in their ecology. There are similar elevations. A lot of the macroecological traits that we can measure are similar between sites. And so all of these shrews were collected throughout the years and dissected in the field, caught with pitfall traps. And those GI tracts from shrews were flash frozen in liquid nitrogen and then deposited in the Museum of Southwestern Biology Mammalogy collection. And so during my master's thesis, I got my hand on these specimens. I sort of unzipped the GI tracts into a petri dish, squeezed all that stuff into some test tubes, smashed them to smithereens, and did some DNA extractions. After that, I did PCR to amplify the 28S RNA gene for cestodes. I used a one-step PCR to tag the products with these specific barcodes so that I could process one gene for all of them, and I sequenced these on And when I received that data back, I formed these single gene reference libraries from morphologically identified cestodes, so sequences from cestodes where we were confident what worm we were looking at. And then I formed a library and searched my results against these sequences that I had aligned. And so what this did was it generated presence-absence data for tapeworms within shrews. And we can use presence absence data through something called the EMS framework, which stands for Elements of Meta-Community Structure. And this looks at these, like a site by species matrix for us, hosts and parasites, and investigates whether or not we can detect coherence or interruptions in the distributions of species, whether or not those species experience turnover, so the number of times one species may replace another between sites, and whether or not those replacements manifest in boundary clumping. So if these replacements happen in groups of parasites or just sort of individually between parasite taxa, and that leads to these two sort of characterizable outcomes that we can think of. So if all of the parasites are responding in concert to shared ecological climes, then we might see Clementsian structure. But if they are responding idiosyncratically, right, each species responding on their own, then we would characterize that as Gleasonian structure. And really, this is just putting a name to the process here. And so you start with your site by species matrix, right? You reorder this matrix using a technique called reciprocal averaging. What this does is it makes it so that observations of hosts, which we're used to calling sites, that are similar to each other in infection patterns are assigned similar ranks or similar score. And so this reordered matrix results in this rank that we can then predict and look for forces that kind of shape this stratification. And so we use boosted regression trees to predict this host rank, looking at variables such as day of the year, host traits, body mass, sex, host species, and environmental traits like maximum temperature and average precipitation. So now you're looking at a site by species matrix where the species here are detected cestogenera, and the sites are each individual host that's on this y-axis here. And so this has been reordered using reciprocal averaging. And the next step is to try to figure out which kinds of mechanisms shape this structure, right? And so we tested, first of all, for coherence and species turnover, which we detected. However, when we looked for boundary clumping, we did not find that to be significant, which we interpreted as Gleasonian community structure, meaning that these communities are responding, like individual species are responding idiosyncratically as opposed to all at once. But the results of those boosted regression trees found that day of the year had the greatest relative influence on host rank. And so but followed by environmental variables like precipitation, host mass, precipitation and temperature. Host mass was also important, but traits like host sex and host species contributed less to host rank. And so since day of the year was the greatest predictor or greatest relative influence on host rank, I decided to check out what that pattern looks like overlaid with my site by species matrix. And I found that day of the year, there are clear gradients of time that are occurring in the cesto communities that I investigated, but I was surprised to see this bimodal distribution here, where the pattern is repeating itself sort of in waves. And I was thinking to myself, what did I miss? I've looked at host sex, I've looked at host species, what's going on? I made some wrong assumptions about how similar those sites were to each other, y'all. Each one of these are the two creeks. But what I found was that these patterns of day of the year being the greatest predictor repeated within each creek. And now I'm also able to see that some of these cestogenera are actually specific to individual creeks. And they also respond, for instance, Staphylocystoides responds oppositely to day of the year, as opposed to this other undescribed genus in Hymenopellidae that has almost an inverse relationship with day of the year. And so this seems like one gradient might actually be really predictive of meta-community structure, but we've detected Gleasonian meta-community structure here, which says that it's actually a bunch of different things. So which is it, right? Gleasonian structure, or is it that day of the year is just sufficient? So I zoomed in on what the kind of individual predictors of parasite prevalence were at the genus level, using the same covariates that I had investigated before in the boosted regression trees. And I found that some of the cestogenera demonstrated similar infection patterns to each other. This is Monocercus, and then this is a cestode that we were unable to identify as a genus, but we know that it's in Dilepthidae. And we see similar patterns in relation to, for instance, maximum temperature here and day of the year. So this y-axis, all you're looking at is just infection prevalence as like considering all other measured covariates. traits. This is just the smooth function on the y-axis. But you see similar patterns here with day of the year. But please note that the scale of these axes are different, and that's important to consider. We found that others didn't. So this Paracyprus and Staphylocystoides has a different relationship with temperature than Mathevolepis. But they both tend to decrease in prevalence with day of the year. We see that this one also has an interesting relationship with host mass. So different responses to environmental and host traits that manifest in changes over time. So if we were to revisit these questions, the first one is, do these cestode communities, do they undergo seasonal changes in species composition? Answer is yes, at least if we're calling day of the year seasonal change, right? And this pattern repeated across both sites.